Hi all, welcome back to Microbiology Lectures, and today I'm going to finish up talking about growth of bacterial colonies and kind of the influence that different things have on the growth, microbial growth. Last time we talked about temperature and pH and salinity and oxygen requirements, and now we're going to talk about nutritional requirements of most populations. And so one of the big things that determines whether or not a microbial population can grow, or at least the rate at which they're going to grow, is the availability of nutrients in a system. And so when we kind of look at the microbial world, or microbiology, and the microbes that might be associated with it, we normally distinguish organisms into kind of groups, typically two main groups. And then there can be offshoots of this groups, and there are offshoots of these groups, and we'll get to that in a second, okay? There are groups which we call heterotrophs, and these are organisms that use what we call organic carbon. And that means that carbon source was generated by another organism. Okay, so heterotrophs, you think of heterotrophs, some way, somehow, they consume organic carbon. Okay, they consume an organic molecule, okay, that was generated by some other organism. Now that could be that they actually just eat something else. It could be that they eat something that's dead. They might have a symbiotic relationship with something that's generating this organic carbon. So there's lots of ways in which this can occur, but a heterotroph means that they didn't make it themselves from an inorganic source. Autotrophs, okay, and this is not just carbon, but when we're talking about carbon, this is what we're talking about. You can have heterotrophs and autotrophs for other elements, not just carbon. But autotrophs, on the other hand, they're using inorganic carbon, okay, to generate energy, and they generate it within. Okay, so they're the organisms that are actually generating. So they're taking carbon dioxide or something related to it. So maybe this is, when we're talking about autotroph in the form of nitrogen, they'd be taking in nitrogen gas, potentially. And they're converting that inorganic molecule into an organic molecule. Okay, so those are the two big groups, heterotrophs and autotrophs. And so when we talk about like humans, we call humans heterotrophs. When we talk about most plants, we call majority of plants, 99% of them or so, are autotrophs, and they're taking in organic carbon. There are a few that don't. Okay. And like I said before, carbon doesn't have to be the only source of energy, or it doesn't even have to be like the preferred source of energy. There are microbes that their preferred source of energy is nitrogen. And so you can have the same kind of classification or group. You can have heterotrophs that use organic nitrogen, so they're consuming amino acids or nucleic acids from outside. Or you can have an autotroph nitrogen, or user of nitrogen, where there might be what we call a nitrogen fixer, or go through nitrogen fixation, where they're converting things like nitrogen gas into a molecule in which they can use. In many cases that might be ammonia, and it could be that they could convert it into potentially into urea or uric acid, but some form of nitrogen, that inorganic molecule is getting converted into an organic compound. Okay? And like ammonia is pretty common, but I want to preface it. It depends on the environment, on how they're converting it. So they would convert nitrogen gas into a nitrite or nitrate, and then the nitrate or nitrite are converted into ammonia. And for a lot of different bacteria, microbial species, that's the conversion pathway. But for some, maybe if water is limited, they might then convert ammonia into something else like urea or uric acid, or some of these nitrogen heterotrophs might be utilizing some of those other molecules like urea, uric acid, or ammonia to get energy from those to then build or use those as building blocks for amino acids and nucleic acids. Okay. There are other things that are often limited and needed in order for the organism to grow. So remember, we're talking about growth. We're talking about how these organisms are basically, they're sitting at that second kind of phase of the growth curve, which is exponential growth, or that log growth. And that's where these organisms basically want to be. They want to replicate. They want their populations to increase. That's their goal. Their goal is to spread their genetic material as much as possible, grow and prosper. And some ways in which they have to do that is by taking in other molecules that might be limited, like phosphorus and iron are often limited nutrients. Maybe potassium, nitrogen is often limited. And we'll talk more about the role that some microbes have in symbiotic relationships. Like a lot of nitrogen fixers have a symbiotic relationship with plants. And so they occur at the root zone of plants, or root nodules. They form these little root nodules, and they might fix nitrogen, and in return the plant might give them a carbon source. So the plant might give them a carbohydrate in return, or they might steal the carbohydrate in return. Depends on the type of relationship they have. But at any rate, there's lots of community effect that's going on when it comes to converting these things. Okay. And we know that growth rate is often directly related to whatever the energy source and the availability of the energy source in that environment. So, as you might expect, if you're looking at a microbe that its main source of energy might be carbon, and you supply it a low, or nutrient concentration is low carbon, okay, you might expect for it to have a growth rate that's fairly slow. And the opposite is true. Like if you supply it lots of carbon and it has the ability to burn that carbon up and utilize it for energy and generate new tissue or new capsule and new cell wall, and they can grow, then you would expect for them to have a high growth rate. Remember, when we're talking growth rate in most microbial populations, or we're talking about growth rate, we're talking about the growth rate of the population. We're talking about cells replicating themselves. We're not talking about growth rate like we would in, say, of a human, where we might be talking about a measurable growth rate of like height or mass, or some of those things. We're talking about number of individuals in the growth rate here. And so the higher the carbon source, often the higher the growth rate. And it's to a point. Eventually you get to a point where you can you can put more carbon in, but your growth rate is maxed. Okay. All right, so other things, there are components, or there are elements of that are out there that are super important for building the biomolecules. Okay, so 4 basic biomolecules. So you might remember these from previous courses, okay, where you have proteins, and proteins are built of amino acids, and amino acids, they need nitrogen, okay? They need carbon. Often they need oxygen and hydrogen and other things, okay? And so those elements that would go into building proteins are a major component, and so those are major elements that determine growth, okay? And so the major elements like carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, magnesium, calcium, and iron, they have a direct relationship with the formation of biomolecules. Proteins, lipids, nucleic acids, and proteins, lipids, nucleic acids, and carbohydrates. I'm sorry. Direct relationship, though, to the 4 biomolecules, the major elements do. So they're often, if one of these are limited, or if some of these are limited, that has a direct effect on the growth rate of the population. There are some trace elements that are really important, like cobalt and zinc and copper, and a few of these other ones that might have a unique function for individual bacteria colonies. Like maybe it's important for the construction of a specialized capsule, or the construction of an exotoxin, or potentially an endotoxin, or something unique. Often we see the trace elements are important for that unique pieces, but the major elements are almost always, these are the limiting sources. So if you want to talk about a population's growth being limited by an element, then you can say, well, oxygen or carbon is one of those big ones. Okay, so carbon, oxygen, hydrogen, again, super important for amino acids, lipids, nucleic acids, and sugars. Okay, and then other things like iron plays a really important role in certain enzymes that, especially there's a couple enzymes that are important for the electron transport chain that and a lot of organisms will use iron. So, but at any rate, just keep that in mind. Major elements are often the limiting source for growth in a population. Okay. All right, so now looking at those energy sources, again, I said you have heterotrophs, autotrophs. We can kind of further break them up or classify them a little bit better. You can have phototrophs. So these are organisms at which they use photons, sunlight, not always sunlight, they use photons, okay? So humans have created artificial light that drives lots of photosynthetic organisms now. And so under normal conditions, they would have used sunlight or the reflection off the moon to drive this energy, okay? Under our conditions, under anthropogenic conditions, human conditions, a lot of these organisms can survive under artificial lights, but they're still using photons. Okay? So photoautotrophs are organisms that are using energy, photons, and some outside carbon source that was not generated or were not created by a living organism. So something like carbon dioxide. Okay? Photoheterotrophs might use the energy from the sun, but they're also using carbon from organic compounds. Okay? So these are organisms that do both. They might use the energy from the sun And then they might consume organisms, or they might have a beneficial organism that's producing some organic compound, or they may produce the organic compound and then utilize the organic compound. So you can have an organism that kind of fits both categories. But photoheterotrophs, normally they'll use the photon to help cleave off carbon or utilize it to generate energy, so they can break down organic compounds. Okay, so we know a lot more as we keep going. We know a lot more about photosynthetic bacteria. Remember, we talked about Lynn Margulis and her ideas that photosynthetic bacteria probably gave rise to the chloroplasts that we see in photosynthetic eukaryotes. And with more and more information and better photography components and better microscopes, we start to see the connections are really there. Like, this is a photosynthetic bacteria. You can see these layers of membrane that they form, so they can capture photons and then utilize the energy from the photon to drive work. So they can pump protons across the membranes and then have them fall back in to generate ATP. And then this is a picture in a electron microscope picture of the chloroplasts of a plant, and you can see these granum or these thylakoid stacks, and they look a lot like those. And so the more information we get, the better the connection is, and you can start to see that whether we're talking about prokaryotic or eukaryotic systems, they're ancient, and how photoautotrophs and photoheterotrophs work is very old. The other option that we sometimes talk about, especially when we're talking about bacteria and archaea, is the chemotrophs. Okay, and these are organisms, okay, that extract energy from organic molecules, okay, like carbohydrates, lipids, proteins, nucleic acids, which lots of eukaryotes fall into this. Okay, but there are special chemotrophs that can extract energy from inorganic chemicals. So instead of using sunlight to drive the formation of, say, a carbohydrate, they use things like hydrogen sulfide or hydrogen gas, and they can drive it. And so we normally call those organisms that use inorganic compounds to form organic compounds, but they use inorganic compounds with the help of energy from a chemical, that we call them chemolithoautotrophs, or chemoautotrophs, or chemolithotrophs. There's lots of terms for them, but they're using chemicals like hydrogen sulfide or hydrogen gas and inorganic compounds like carbon to form things like glucose or sucrose or some formation of a carbohydrate. Okay, so instead of using light, they use chemicals. We are also, humans, and lots of eukaryotes, are also organoheterotrophs. So we don't use chemicals to make carbon-based products, but we use chemicals to break, or we use the energy from the breaking of these organic compounds to drive work. So we're often called chemoheterotrophs or chemoorganotrophs, or chemoorganoheterotrophs, because we use energy from carbon, or from nitrogen-based products, or from proteins, lipids, nucleic acids, and carbohydrates. We use the energy from those to generate other compounds, or to use the energy to drive work. Okay, so with that kind of wraps up kind of the connection between energy and growth rate in these different organisms. And Now we're going to switch gears just a little bit, and we're going to talk about how can we control growth in different situations, whether that be in the home, medical facilities, all over the place. All right, next time.